Hello, everyone. Welcome back to the podcast. I'm Yathika Sivanimuthakrishnan, the CEO and the founder of the Brain Camaraderie, a youth-led nonprofit dedicated to supporting patients impacted by rare neurological diseases.
And I'm Christine, the communications lead at the Brain Camaraderie. We share stories, research, and action around rare brain diseases. And I'm thrilled to be here today with Yathika to co-host our second podcast episode. Today, we have the honor of speaking with a true pioneer in the world of neuroscience and genetic medicine, Dr. Ralf Schmid. He serves as the Associate Director of Preclinical Research at Novartis and has dedicated his career to advancing breakthrough therapies for neurological conditions. Ralph has worked on gene therapies and gene editing approaches for diseases like Rett syndrome, ALS, and glioblastoma, and his team has successfully used CRISPR gene editing therapy in the Angelman syndrome mouse model.
What makes this episode so special is that we'll be looking at how cutting-edge science interacts with children's lives, ranging from rare diseases in babies, to the future of gene therapy, to those ethical and social barriers children face.
Dr. Ralph, thank you for joining us. Can you kick off this podcast by sharing who you are, what you do, and what drives your work?
Well, thank you very much. It's really a true honor to be part of the series of podcasts, and I'm happy to spend some time with you today. I was trained as a biochemist. I spent a lot of my professional life working in neuroscience, which is the science of how the brain works, and the associated neurological or brain diseases. I spend a lot of time in academic institutions at universities doing research into those brain conditions, and more recently joined a large pharmaceutical company to try forward some of those findings and which hopefully ends up becoming some therapies in the future for those brain diseases.
Thank you, Ralph, for sharing that with us. You've dedicated your work to the brain, which is one of the most difficult parts of the body to treat. Can you walk us through why neurological diseases in children are especially hard to diagnose and treat from a scientific point of view?
Yes, so neurological diseases in children manifest themselves um, often very early. A lot of those diseases are of genetic origin. That means already in the womb and soon after, the brain is not functioning exactly the way it should be. So unfortunately, very young children can't really talk or explain exactly what's wrong, and so it's often discovered when those children miss milestones, so that they fail to appropriately move at a certain age, or then start talking or responding to any kind of cues. But then again, this is very non-specific, and often then a long journey starts both for the patients and also the pediatrician to find out what exactly is wrong. And sometimes that can take really, really long. And so in this regard, genetic testing of a child displaying some kind of symptoms or concern can really help to narrow down or even to pinpoint it to a specific genetic disease, which then in turn can facilitate coming up with solutions with therapies.
Thank you for explaining that detail. And for those of us getting into neuroscience, could you explain what the blood-brain barrier, or the BBB, is, and why it makes delivering treatment to the brain such a challenge?
The blood-brain barrier is some kind of safety switch nature invented to basically isolate the brain from the rest of the body, so that whatever you eat or drink or in other ways gets in your blood, that doesn't hit the brain immediately. And so that's necessary, for example, to keep bacteria, virus out of the brain, limit the access of certain other chemical substances to the brain on one side, but on the other side, it also makes it very difficult to get medicines in the brain, because Uh, Medicines, drugs, pills, any kind of injectables underlie the same restrictions to enter the brain. And the blood-brain barrier is really a very specialized surface, what we call a specialized surface. So it is between the bloodstream, the blood capillaries, and the brain space, and it's a very tight line of cells which are tightly policing what they let through and whatnot. And so this really has been from the beginning of treatment of neurological disorders a very big obstacle to design not only a medicine, but design a medicine in a way that it actually enters the brain um and treats successfully treats whatever is wrong with the neurons or the nerve cells in the brain.
Thank you for sharing that with us. And what innovative strategies are being developed to cross that barrier and deliver treatments effectively, especially to a child's brain?
Well, fortunately, it's a general mechanism. So whatever typically solutions are found for adults would work also for children. Um, we But I have to say we don't know that much about the blood-brain barrier in very young children, in newborn. So the thinking is that the blood-brain barrier is less formed, and so that's why it should be a little bit easier actually to get drugs into the brain of very young children. But in general, the strategy is to modify drugs in a way that they can pass the blood-brain barrier, that they can use a transport mechanism, which are the so-called good substances. So, for example, glucose or other nutrients would use to pass the blood-brain barrier. And there's a number of receptors on the blood-brain barrier. They can be utilized to get either drugs or antibodies or even viruses to deliver gene therapies across the blood-brain barrier and successfully inside the brain.
Thank you for sharing that, and thank you for sharing how, um, and thank you for sharing about the transportation stuff too. Many children with rare neurological diseases go undiagnosed or misdiagnosed for years. So, from your perspective, what are the biggest obstacles of getting an early and accurate diagnosis?
Well, as I mentioned before, the symptoms are normally non-specific, and the children really can't help with the diagnosis. So, if you have somebody who might have Alzheimer's or not, you can run certain tests on them. You can do cognitive tests, learning and memory tasks, and then figure out really where the problem lies. So unfortunately, we can't do that with children until a little bit older. And so in this regard, you can do all kinds of imaging to exclude possibilities like having a brain tumor or any kind of hemorrhage or any other more physical problem with the brain. But then, it's often just the neurologist, the pediatric neurologist will conclude, well, there's something wrong with the brain, but we're not exactly sure what. And so there really, what can help is a molecular diagnostic. So a diagnostic which is based on sequencing the genome of the child and determining if there's any known pathogenic, what we call pathogenic or disease-causing mutation. There are catalogs of disease-causing mutations, um, uh, and the associated genes, and then the associated symptoms, the associated diagnosis, the associated syndromes which go with that.
And how could genome sequencing as a part of newborn screening help solve that problem? Is it realistic that one day each child might have their DNA sequenced early on?
Well, that would be great if we could accomplish that. Um, that is probably, as I see, the most accurate way to really determine the disease a child has. The problem is just, a lot of those neurological diseases in children are rare diseases. So, they might occur in any given hospital, depending on the size, maybe a couple of times per year, maybe every couple years only. So it would require really to screen pretty much all of the newborn babies, or at least any of the newborn babies which have any kind of glimpse of that something might not be completely okay with them. And so that is quite a large number. The technology is still quite expensive. So just the network of sequencing facility, genetic counseling is not really fully built out yet. So it's mostly restricted to large medical centers, to bigger hospitals, to teaching hospitals. So there's a lot of work to be done. So there are several initiatives right now going on, pilot initiatives. And so I do hope that in the future that more hospitals can participate in this kind of screening, and just that it becomes routine screening. Typically, a few drops of blood are taken anyhow from babies for some of the older metabolic tests to figure out at least if more common metabolic disorders are not present in the child. And so it would be great if also a few drops of blood could be used for sequencing of any given child in the future.
That would actually be a great initiative. It would really help with earlier diagnosis. And could you share a story or a real-world example of how earlier diagnosis could have helped or has helped a child that you've worked with, while respecting patient privacy, of course?
Yes, a couple of years ago, I was introduced to a family with a at that time already teenage daughter who had a genetic neurological disease, but that it took really long. It took about 14 or 15 years to get a final diagnosis. So the child was born, and first it seemed to be development seemed to be fairly normal, but after a year or two, the little girl was falling behind in milestones. I mean, she had just trouble the normal crawling, standing up, forming words, making use of hands, learning fine motoric skills and things like that. So clearly something was not okay. And so for the parents and their daughter, it was an insane journey through dozens of different medical offices, hospitals, and really nobody could figure out what was wrong. And the final diagnosis was just, yeah, low IQ. So something wrong with the brain. She'll never grow up, and the brain will never learn. And it really, just by chance, they learned about an initiative, about a sequencing initiative, and that was already a few years back, when a company was looking for volunteers because they were trying to figure out those services. And so they provided a blood sample, and then it was really surprising to the parents when the company said, "Well, we found something. We found something, which is clearly not present in many other children. It does not match the human genome, which is posted in the databases. There's one particular mutation in a gene which has been associated with a brain disorder, in girls." And so that was, and the problem also was, it was a mutation which really had not observed before. So at that point, that girl was the only known patient in the whole world with that particular mutation. I think since then, two or three other girls have been found to have the same mutation. So now there's actually an entry in the disease in ClinVar, which is a database of mutations which cause disease, that this particular mutation is associated with a brain disease. Unfortunately, there's no cure for this brain disease, but it at least helps then to connect with other parents, to connect with patient organizations, and just try out what other parents have tried to at least ameliorate some symptoms and help the child to certain extent.
Thank you, Ralph, for sharing that story with us. It's it's when people often just stop at the first step when doctors just say, "Oh, it's low IQ," they just refuse to look further. Do you Has there ever been an occasion where people thought it was a mutation, but it was just a regular um, delay in like achieving the milestones?
Yes, so that's a little bit the problem with molecular diagnosis, that we all differ in our genomic setup. It's like 99% it's the same, but then among those trillions of bases, of DNA bases we each have in our cells, there are certain variations. So a lot of those variations, they encode, what kind of hair color, hair texture, eye color, how tall you get, if you're born to be a marathon runner or rather a chess player. So those things are normally encoded somewhat, but also, there's also things encoded, for example, if you're lactose tolerant or intolerant, or if you have any, or some other more serious allergies can also be part of your genetic setup. And so it's really difficult to sort through that. So you really can only build confidence if there's already many other patients who have exactly the same mutation in the exact same gene. That would build confidence that you have that disease. So what normally is done, so if a mutation has been found, you know, the genetic testing service, they would narrow down on that particular disease and then ask, what are the related diseases here? So where it's a different genetic reason, but kind of the same symptoms, and then look really careful at those other genes which can cause similar symptoms to really rule out that they're looking at the wrong gene here, and so further confirm the diagnosis. But yes, of course, it can still happen that it says, well, there is some mutation or there is some abnormality in the gene. We don't know exactly what it means, but let's just wait. And so either then the child gets worse, or as you said, it's just a normal delay that some kids just need a little bit longer to meet the milestones, and everything is going to be fine in the long run.
Thank you for sharing. And I feel that it's a very lengthy and complicated process, so I really only hope that it gets
Definitely.
easier in the future. Your work includes creating personalized gene therapies using CRISPR and other tools.
Yes.
So, could you explain how this technology works in simple terms for our audience?
Well, this is something quite new, and so some of the older listeners might only have heard about this in the news because, when you were in school or in college, it simply didn't exist yet, so there was nobody who could teach you that. Um, so the CRISPR technology is something very smart scientists originally discovered in bacteria and then found a way to redesign the system that it would also work in mammalian cells. It means in any of our cells. Um, and it is a way really to change the genetic code. So as you know now, normally all our genes follow a certain code, which is made out of 4 different base pairs, and that's everything in our body is basically encoded. And so now the disease-causing mutations is often it's only a one base pair, a one letter difference. And so this technology now can be used to edit the gene or the DNA sequence, which means you make one little change, and often not much more is needed than one change or maybe two base pairs of changes. So those it's a combination of different enzymes, different molecules, which can be designed in a way to hone in on a specific DNA sequence, look at the DNA sequence, find the mutation they were trained or they were designed to correct, and then settle down on the DNA sequence and by a number of enzymatic steps excise the wrong base pair and replace it with a correct base pair. And so that's, in the beginning, this was very inefficient. So it worked sometimes, but not often. And sometimes also it made mistakes. It replaced the wrong letter with another wrong letter. So that's not something we want. It took many years to design even more complicated systems now, which have checks and balances built in, where there's basically a second module which looks at the DNA sequence again and verifies that the correct sequence is there now, and then disengages or kind of instructs the enzyme to cut again and try again. And so I think we are finally at the point where those gene editing systems are accurate enough that they can actually be used in a therapeutic setting for human disease.
It's amazing to see how much technology has changed to be able to access the most smallest part of our human body, like the cell, and then figure out DNA and to modify the gene so that the mutations don't affect us anymore. Ralph, you were part of a team involved in treating the first patient with a personalized CRISPR therapy. What did that moment mean to you, and what could it mean for children with rare diseases into the future?
Sorry, we need to cut that out. So you didn't get the change. So I was actually not involved in in that. So I'm not sure, Yatika, if you have a reworded question or if you want to do that real quick. It's number 9. It's at least on my script, it's okay.
Wait, I'm so sorry.
Yeah,
That's my
no,
fault.
that's fine. So just start again with that question number 9.
Okay,
All right.
Chris, give me a minute. Let me change it. So we have like two scripts, so
Oh,
that's probably why we were struggling a
I
little bit.
think that's the only one.
So Chris, do you see the document? It's this one, this question. Okay, we can start again.
All right, I see it. Thank you so much. Dr. Ralph, what did you feel when you first heard about the world's first personalized CRISPR gene therapy for a patient?
Well, this was really, this was really exciting. This was really cool news. I was actually I was at a conference, and that came out of nowhere because typically those big announcements, they're shrouded in secrecy, and actually until the actual event began, nobody except the organizers knew about this. And I think everybody was completely astounded. People got incredibly excited if you heard about that, because what they actually did, it was a child in Philadelphia in the Children's Hospital with a very serious metabolic disease. So they were able to ameliorate the situation in the child by nutrition, but typically those with that particular disease, the life expectancy is if the child would be lucky, is maybe a couple of years, but often it's a lot less. And so what they were able to do is not only find the defect, find the mutation, but then design a personalized therapy within just a couple of months, essentially. Successful perform the gene editing, and then at that point, when the announcement was made, they were actually starting to wean off the child of the special nutrition and switching over to just regular formula. And it seemed like that the child was tolerating normal nutrition very well. And the expectation is, that the child will have a normal development and hopefully a normal life.
Okay, thank you. And like you said, the therapy has to be personalized and it has to be tailored to one child's specific genetic mutation. So, can you tell us more about how this therapy is designed and the process, risks, and challenges involved?
Yeah, so with gene editing, that's one of the downfalls is that gene editing will always address a specific mutation. But as I mentioned before, a lot of diseases, including neurological diseases, uh, it's one gene is to blame, but there might be 100 ways to screw up that gene. So there's 100 ways to uh, where the mutation could be, how the gene gets truncated, it's not functioning correctly, and so the trick normally is we're trying to find therapies which would help the vast majority of the patients. And so that has been in the past more like if you think about pills or something like that, they're typically addressing more the symptoms, the underlying defects, but not the genetic origin, which is, one step further. If you fix the genetic origin, really you don't need to take any medicine anymore for the rest of your life. You don't need any other therapies. So now the trick is really to, how are we going to do this? It sounds kind of counterintuitive to start with one child with one specific mutation, but that's actually a good way to figure out how those technologies work because it's very defined. It is one patient with one mutation, so you can exactly study the outcome and then try to, the future step would be how can we generalize this to other children with the same mutation, and then, you know, same disease but other mutations. Can we kind of roll this out for different mutations, different diseases in the end. And so what that really requires as the first step is sequencing so that you're absolutely sure what the mutation is and to ascertain that there's only one mutation. So sometimes we also have children, they have what's called mosaic. So they could have different mutations or, some cells have the mutation, some not. So you have to plan carefully that your therapy is actually addressing the problem. So once you know exactly what you need to change, then you need to design your tools for that. And so the good thing is that I mean, the vast majority of the gene editing complex is reusable because those are enzymes which cut the DNA, which repair the DNA, which put the correct base in, and so on. But what is individual, what needs to be designed is actually that part of the CRISPR complex which finds the mutation in the first place. And so that is done by designing a small piece of DNA which matches exactly the one piece you want to change. And so that is really so that is the first step, that you design this and you test this out on cells, that it really finds the mutation, and then go through the entire process, basically put all the pieces together, try it first in a cell model. In this case, they had to create a mouse model real quick to fully test that gene editing, because often you can only, if you treat an entire organism, you can see that it actually solves the problem and has no severe side effects. Um, After you test that in an animal model, then you're pretty much ready to go. There's a few other things you can computationally uh, model some things, look at side effects, investigate side effects. You would need to figure out, like what kind of dose would we need here. So kind of calculating using models to go from animals towards a patient. In this case, they were very conservative because it was the first time, so they actually did the same editing 3 times to make sure it really worked. So they used really low doses to not overwhelm the body, and used a very small dose, and then increased the dose a little bit, and then increased the dose even more, and so they think they ended up at very efficacious editing. So they opted not to look because, doing in this case it was liver directed since it was a metabolic enzyme. So it was part of the liver was edited. You could theoretically, just go in, take a liver biopsy, and verify that everything is okay. In this case, they opted not to do this because it was a newborn baby. So, whatever you can avoid doing to that baby, there was just no justification. So maybe in the future they will look actually and see that really how successful their therapy was. But that is in a nutshell what you would need to do every single time. So the hope is that we can streamline that somewhat in the future so that it becomes a lot more modular. That there are already cell line models there. There is somebody has worked out a protocol instructions how to make this complementary DNA pieces for searching the mutation. How would you go along with doing the different testing in cell line models and animals? How to do all this dose selection, de-risking, potential safety risk, and so on. So that is the hope that in the future that we that researchers and pharmaceutical companies in collaboration can figure out how to exactly do this, and could become a lot easier, hopefully one day.
Thank you so much, Ralph, about for sharing that. You spoke about babies, and I'd like to ask about the ethics, especially with limited resources and having to create a specific segment of DNA for separate mutations. How do we decide who receives these powerful but limited therapies first? You meant
Yeah, so that's a very good point. So I want to emphasize they're still all experimental or investigational therapies. So it's not something you can just go and place an order in the hospital pharmacy and then get an injection the next day. It is really, it is being approved by the Federal Drug Administration or FDA on a case-by-case basis, for now, because it is still in the early phases. So I think we can all agree on that, severe disorders which would severely restrict the life expectancy in an otherwise healthy child is clearly something, um, which requires our attention. So I think a lot of times, like in this case with a baby in Philadelphia, there was not much what anybody in the hospital or any doctor could have done for that baby, except it would the child would have died pretty soon because of the severity of the disorder. So I think there it's easy to say, we need to try something, and so we need to figure out something to help children like that. So it becomes a little bit more complicated. Of course, the risk. There's a risk because it has not been tried before. So the parents were aware of that there was a risk, but they wanted to take the risk because they put all their hope basically into that this novel therapy would actually do something good for their child. It could have gone awfully wrong, obviously. So yeah, so there's a risk, there are ethical considerations, which brings me to the second point that in this investigational experimental status, you have to think carefully about the kind of conditions you might prioritize for treatment. And it really, what we call, it's the benefit risk ratio. So, if you have a high risk, then there has to be a tremendous benefit. So in this case, survival of the child was the benefit. It becomes more difficult if it's a milder disorder, which, yeah, it provides discomfort, might rob the child of normal development, but then at this point, it's often that ethicists and there's always ethicists involved in those decisions. Together with the parents and the doctor might decide, your child really doesn't qualify at this point. We hope in the future, once this has been all worked out, and there's multiple examples of how this works, that it could be widened to a variety of other disorders. But I think this will take it might take another 5 years, 10 years. I don't know. So you really need to have a large enough number of children where those personalized therapies have been tested on.
Thank you for sharing me the efficacies, and I completely understand it's really uncertain out there, and it could take a very long time. Let's shift gears a little bit. Can you tell me about the stigma that surrounds disorders now, especially in the underserved and religious communities, and how they may actually impact the children?
Yeah, so this is something, especially in the neurological space, we're all still struggling with. So, ideally, we would like to go out, diagnose the children, deliver the diagnosis, and then work together that clinicians and parents and all kinds of other caregivers would work on a therapy for the children. But it is still, I think, neurological disease is still sometimes seen different as something else. So like we've made great strides in the acceptance of, for example, like different forms of cancer or dementia, like Alzheimer's disease, where I think everybody is open to also talking about it, seeking help. Versus, if your child is different than other children, it's sometimes it might be also a little bit a cultural thing that you don't want to talk about, that you're trying to kind of hope that it's going to get better, kind of hide the fact that there's really really wrong with your children. And so that's very difficult to kind of educate all of us that really, the brain is just another organ. I mean, disease can manifest itself that you have a tumor, that there's something wrong with your heart, something wrong with your liver, but it could also be your brain. It's just that typically brain disorders, they manifest themselves often in a more obvious way. So it could be in forms of epilepsy, or any kind of episodes, any kind of episodes, difficulties to express yourself verbally, difficulties to use your hands or your arms. And I think there's still a lot of education needs to be done, that they are, those are just diseases as any other disease. And yes, there are a lot of people working on therapies for those diseases.
Thank you. Could you share a little bit more about like the differences? You said how children with neurological diseases actually get treated a little different than maybe with children with cancer. So could you maybe elaborate more on that?
Yeah, I think one thing probably is also due to the fact that for many other, or let's say organs, for many other parts of the body, therapies have been identified and successfully used in many, many patients. So, you mentioned cancer. So there's pretty much for every kind of cancer, there's a therapy. So sometimes, it is very efficient and the cancer doesn't come back. Other times it's unfortunately not as successful. But I think, most people would probably, if you get diagnosed with cancer, your first question is, so what are we going to do about this? um, and often the doctor can give you an answer, or can send you to a center which specializes in that particular cancer. So unfortunately, with neurological diseases, it's still a lot more like, well, it could be this or it could be that. So, and then even if you get a diagnosis, if you have a molecular diagnosis and you know exactly what kind of syndrome your child has, a lot of times, it's still, well, I can give you a brochure, but there's not much else what I can do. And you just need to connect with other parents, or I can send you to a physiotherapist or to a nutritionist, and they can help you. But it's still very difficult, and I think it's also very difficult for the parents to accept that there's something seriously wrong with their child. Also, just to live with that, because it's an immense burden on the parents, on the caregivers, on the rest of the family. If there's a child which never really gets out of that toddler stage where they need 24/7, where they need somebody providing care for them 24/7.
Thank you for sharing that, Ralph. You also spoke about the stigma that surrounds neurological disorders, especially in underserved or religious or even specific places like demographic variation. How does this impact children, and what role can education play in reducing that stigma?
Chris,
We
we
got
already
that already.
we already
Sorry.
asked that.
Um, do
Um,
you?
uh,
I think we covered that. Do you want to move on to the next session, to
yeah.
section 5, maybe?
Okay, Chris, we already covered that.
Yeah, yeah.
You can start with
Well, it's more
uh,
that we get to your role in the brain camaraderie and things like that.
it's okay, we can cut that part out, so please continue.
In Brain Camaraderie, we believe patient voices and stories should be heard in research process. How did these stories and shared data influence your research?
Well, the more we know about diseases, but not only the textbook knowledge, or in this case the internet knowledge about what the symptoms are and what the cause of disease is, but actually hearing from patients or their families is very important, because we learn I mean, I learned a lot about pediatric neurological disorders that way, about different disorders, by just talking to families or talking to patient advocates. You get a much better sense what the problems really are, how urgent the needs are, and how neglected some of those patient communities feel, and neglected in a way that there's just not much you can do. There's clearly no efficacious therapy available. And so it helps us both, to motivate us, to provide us a background, to do the research, but also to better understand what we're really trying to address here, what kind of symptoms we are trying to address, which is very helpful in, thinking about how to design a clinical trial and so on.
What role do youth-led advocacy groups like us play in accelerating research and awareness?
Well, I think this is all, as I mentioned, this is all very new. A lot of those genetic technologies are very new, and so I think the awareness in the general population, on average, is still rather low. And so, as we said, the neurological diseases, a lot of them affect babies. And so I think it is very important among young people also to learn about this, because, I mean, some of your listeners might even have somebody in their family, might have a younger or older sibling who has been diagnosed with a genetic neurological disease. I mean, a lot of you will one day be in the position where you have your own children. And so I think it's just important to know about these things, but also important, we need young people who are willing to train, either as a researcher, as a clinician, or as a nurse, as a genetic counselor, as an engineer to come up with novel solutions. So that's also very important, and we definitely do need advocates. We need voters who also, voice their concerns that there is enough support from the government and government agencies to do research, to develop medicines, to have enough resources that families have access to medical care, have access to the diagnosis we talked about.
Thank you. And if someone listening today wants to pursue a career like yours or do something related, what do you think they should study and learn and be doing right now?
That's a really good question. So, well, first of all, don't be scared if this sounded kind of complicated, what I described here. Yes, it is complicated, but it's also complicated to build an airplane. And there's plenty of you, aspiring engineers who will learn how to build an airplane. So it's kind of the same thing in biology. As long as you're curious, you're willing to really, roll up the sleeves and get really deep into science, learn different aspects of science. So it's not only one discipline. It's a variety of different disciplines, different expertises which are needed. So you might need somebody who knows everything about DNA, who can engineer and make DNA molecules. You need somebody who makes the enzymes. You need somebody who knows how to test therapies on animals. So there are a lot of different occupations, different roles out there which can contribute. And so I think, as a lot of you probably prepare for college quite soon, just be open-minded, look around and try to figure out like which part of this large scientific enterprise, biomedical enterprise is something you're most interested in, and also your skills most align with.
Thank you so much, Ralph. What is the biggest breakthrough or milestone that you hope to see in pediatric neurology in the next 10 to 15 years?
Well, I hope that a lot more what I would call real therapies are approved. So therapies which not only somewhat alleviate the symptoms, but really, remove the cause of the disease, be it either a genetic therapy or some other kind of therapy. So I hope we will see a lot more excitement here of breakthroughs, but I also hope, that innovation doesn't stop. So that there's a lot more things, a lot more therapeutic avenues, discoveries are being made where we really at one day we understand better how the brain works and how a disease affects the brain, but also how just to coming to a full circle what we discussed at the very beginning, how can we effectively treat the brain? How can we effectively get therapies in the brain? And so I still hope, this whole somewhat black box called blood-brain barrier that uh, within the next 10 years or so that we develop a much better understanding, develop much better tools to get our medicines across the blood-brain barrier.
Thank you. And if you could leave our audience with just one really powerful message about children and genetic medicine, what would that be?
Well, don't give up hope. So, it might look like, oh my God, my child has a really serious disease and there's nothing, there's no good therapy. So first of all, I mean, we have made huge progress over the last decade, over the last couple of years to treat neurological diseases, to treat genetic diseases. And then also, you don't, what is my belief, it doesn't have to be perfect. It doesn't have to be perfect. The brain is what we call a very plastic organ. So plastic means it can adapt. So the brain is very good to adapting to bad situations. I mean, just think about having a stroke. So there's often a stroke is when the blood supply is cut off and part of the brain actually dies. I mean, there's dead areas after stroke often in the brain, which is severely impacting how the brain can function, how the brain can control other bodily functions. But then you know, the brain often finds ways to work around this problem. I mean, just to, work around the dead areas, somewhat recover functions, retrain other parts of the brain. And so I think the same thing. I'm very hopeful is true for brain diseases, that we don't need to fix every single aspect of a neurological disease in every single part of the brain. So if it's a reasonable approach affecting a reasonable number of cells in the brain, then giving it some time, giving it supportive care, which could be physiotherapy or customized intellectual stimulation, into learning and memory games and things like that. That I really think a lot of the therapies which are right now in the pipeline are being investigated could really become very successful, in that way.
All right. Thank you so much, Ralph, for your time, your insight, and your dedication to helping children and families around the world. This conversation has been such an eye-opener, and it's got me fascinated, honestly.
Well, thank you. It was really fun talking to you.
Thank you. And for everyone listening, this episode will be available soon on Spotify, Apple Podcasts, YouTube, Podbean, and even on our website, nonprofit website. So please share it with your friends and your community.
And follow us on Instagram, LinkedIn, and Facebook to stay connected with the Brain Camaraderie and to support our work. Bye!
Bye.